Good afternoon, everybody. Like my colleagues, I'd like to thank the organizers, so Sarah and Florian, and also the BrightFocus for putting this amazing pre-conference meeting. I've learned a lot. I don't have anything to disclose. So this is the outline of my talk. I'm going to mainly be talking about what is autophagy, a little bit of history and cell types. How can this process be measured? And then I will go to try to understand how cargo is selected, and we'll talk a little bit about selective autophagy. I will be focusing mainly on mitophagy. I will also mention the best tools to look at mitophagy, and then talk about a couple of examples from our lab. And then, of course, I'll go to the unanswered questions that I was requested to add, and then further reading. So a little bit of etymology is very easy to understand the topic. So autophagy comes from the Greek, which means self and eating. So this is an intracellular pathway that occurs in all of our cells that is used to degrade and recycle intracellular components. And I like to talk about this process is the story of two Nobel Prizes, because many years ago, Christian Ledoux discovered the end part of the pathway, which are lysosomes, and he discovered lysosomes by doing electron microscopy on glucagon-treated rats, and he observed these two types of organelles. The first one, they had both of them have double membranes, but the first one, the interior of these organelles was very similar to the cytoplasm, and in the second one, it seems that the material was under degradation. So just by looking at these two different types of organelles, he was able to think that actually the cell was digesting itself. And then a bit, some years later, you probably remember Yoshimori Oshumi was awarded of the Nobel Prize in Physiology of Medicine because he discovered the molecular regulators of this pathway, which now we call the ATG genes. And he discovered these ATG genes by working in yeast. He did some mutants and found out the molecules or the genes that were important for this process of autophagy. And just to give you an idea why this was so important, this is the number of publications in PubMed during the year. So it was really very low and very stable until this discovery of Yoshimori Oshumi. And then you can see, bam, right away the number of papers in PubMed went really up. And I stopped counting in 2020 because I got tired. But you can see here how important, because then we could have we could manipulate those genes and then see the consequences for cells and tissues. And we now know that autophagy is very important for many diseases, of course, neurodegenerative diseases, and also cancer and infectious diseases. And this line here is just to tell you how old I am, because I've been working in this process before this molecular area, actually. So a little bit of these types of autophagy. So actually, this is we classify those depending on how the material reaches the lysosome, which is the end part of the process. So macroautophagy, which is the most known type, the one I'm going to be talking about mainly today, it starts with the formation of this double membrane that grows and closes to form the hallmark of autophagy, which is this autophagosome. And then this autophagosome will fuse with the lysosome. The second type of autophagy, which is much less known about, is microautophagy. Here, the components to be degraded get directly into the lysosome via an invagination of the lysosomal membrane. And we still really don't know many about these components of this microautophagy pathway. The third one is chaperone-mediated autophagy. This is a selective autophagy which only degrades proteins, because, by the way, during macro and micro, other things besides proteins can get degraded. So in chaperone-mediated autophagy, it's called chaperone because it's a chaperone that recognizes a specific motif in proteins and then helps to deliver this protein to the lysosomal membrane, and there interacts with a receptor which is called LAMP2A, and this LAMP2A will dimerize, and this will allow the unfolding and internalization of the protein inside the lysosome. I'm not going to talk about anything about CMA, but if you are interested in this process, come to the poster 171 because we have a lot of nice data on AMD about chaperone-mediated autophagy. So once the material gets to the lysosome, the important thing that this material gets degraded, and more importantly, the components after this degradation, so the amino acids and the nucleotides and the lipids, can get out of the lysosome, and they can be used for the cell to recycle and to be used for other cell functions. So this is just a picture I just put to make it very visual. So actually, autophagosomes are the recycling track that takes the material to be degraded for degradation. So if you want to know a little bit about the molecular mechanism of this process, I didn't put you this video, but some years ago in the frame of this European project, we made this video, which is 9 minutes, and I say, I'm not going to use 9 minutes of my talk, but you can just go and have a look at it, because there are many of the molecular pathways that are activated and how it works are very nicely explained with this video. Okay, so all cells have certain levels of basal autophagy, so we need this basal autophagy to keep our neurons clean. But of course, this is a stress response, so it's a process that gets induced after many conditions, and the most typical one that you probably all know is starvation. Starvation is the most classical way to induce autophagy. So actually what happens is that when this process is activated, even under basal conditions, these are the cargo that can be degraded via autophagy. For example, you can eliminate organelles that you don't need anymore. And the most typical or most classical way to look at this is when you don't need ribosomes. They are going to be degraded via ribophagy. Of course, you all know when mitochondria get damaged or when the plasmic reticulum get damaged, it can also be degraded via autophagy. And of course, it's very important to eliminate protein aggregates. Autophagy is also very important to degrade intracellular pathogens, and we know, as I already mentioned, is a very important metabolic adaptation that allows the cells to survive under starvation. So the cell uses parts of the not needed parts to degrade them and obtain energy. So when looking at all these important functions of autophagy, it's logical to think that alterations in this pathway can lead to many diseases. So how can this process be measured? So I'm going to tell you some of the tools that we can use, that we use in the lab. And before that, I'd like to give you a little bit more of insight how autophagosomes are formed. So actually, to get an autophagosome, you need two conjugation reactions that are mediated by ATG7. The first one is the conjugation of ATG5 and ATG12, and the second one is the conjugation of phosphatidyletanolamine residue into LC3. And you need these two reactions in order to get this autophagosome formed. So I like to make attention that an autophagosome is the formation of an autophagosome is a very fast process, and it takes around 10 minutes. So you have to think that when you design your experiments, you have to keep this in mind. And the important thing is that you have to measure what we call autophagy flux. And I like to make this analogy for autophagy flux. So imagine it's a Friday afternoon, you are in the lab and you want to go home, and then you want to see whether it's a traffic jam. And then you look there and you see cars. But actually, looking at the numbers of cars out the window doesn't tell you anything whether there is a lot of cars, a lot of traffic. So what you need to do is to put a traffic light and see how many cars are accumulating. So this is exactly what we have to do to measure autophagy flux. We have to put a traffic light, which in this case is lysosomal inhibition, and then we can compare how many cars we have with and without the traffic light, and then with this you can calculate how much flux, how much traffic you have. So it's important to measure autophagy flux. So some of the classical methods to look at autophagy is electron microscopy. For example, this is a very amazing image that you can get from textbooks. Actually, to get these images is very difficult because, as I tell you, it's a very fast process. Here you see in the first stages of autophagosome formation, this phagophore, this double membrane, and you can see here a beautiful image of mitochondria inside an autophagosome. So you have already seen these two images of autophagosomes and autophagolysosomes. The most important thing is to check that they have double membranes and that the interior is similar to the cytoplasm. And then you also could check for lysosomes. And I like to put this image to compare the sizes of an autophagosome with the lysosome. You can see the lysosome is much more smaller. And actually, lysosomes are very heterogeneous. So beautiful images, but they are difficult to get. And also it's complicated to interpret because I told you about double membranes, but you can get double membranes in some other places. For example, endoplasmic reticulum, some of them are also double membrane, and also mitochondria also have double membrane. So sometimes it's a bit difficult. So how you can get these beautiful images, but it's difficult and you need a lot of expertise, and also you need to give numbers. So actually, we don't use electron microscopy in order to really measure autophagy. So the most classical method that we use to measure autophagy is we do the LC3 lipidation by Western blot, looking at autophagy flux. And I have to mention here that LC3-1, which is the non-lipidated form, migrates a certain speed, but then when this LC3 gets lipidated, it's more hydrophobic, and then it can go faster in a gel. So when we do a Western blot, here you can see some MEFs, and then this is starvation, so hours of nutrient deprivation, so half an hour, 2 hours, 4 hours, 6 hours. So this is LC3-1, the form that is not in autophagosums, and this is LC3-2, so this is the form with autophagosums. And here you can see very interestingly that by half an hour, you already have a lot of autophagosums, and if you continue the kinetics, you slowly getting less and less. So again, looking only at these time points doesn't tell you anything about the flux. So what do we need to do to measure the flux? So you have to incubate your cells plus minus protease inhibitors, and then you can see the flux. And actually you can compare the number of autophagosomes, so LC3-2, without and with protease inhibitors. And you can see that in this second case, there is much more accumulation of this LC3-2 band. So this is exactly what is autophagy flux. We need to compare plus, minus protease inhibitors and check how these autophagosomes accumulate. Some examples to make this clear. So for example, if you look at here without protease inhibitors, see this in this same line, you can see that with starvation, you get a little bit of LC3-2. And however, you compare the same incubation with starvation. In this case, in this cell line, you see a decreased levels of LC3-2. So looking at this doesn't tell you anything about the flux or how much autophagy is there. However, when you do the protease inhibitors, you can see here that you have some increase with protease inhibitors and much more increase with protease inhibitors. So this is telling you about the flux. And here, looking only at this band and this band will be confusing, but when you have the flux, when you have the protease inhibitors, you can see really that in both cases, under nutrient-rich and starvation condition, you have autophagy flux. So some of the protease inhibitors or some of the lysosomal inhibitors you can use, the most typical one are protease inhibitors like leopeptin or E624D of pepsidin A, but you can also block the lysosomal pH, which also block degradation. And for example, it's very typical to use bafilomycin or ammonium chloride. And for example, in our lab for the retinal explants, we use a combination of ammonium chloride plus leopeptin. Another way to look at this autophagy flux is by tandem reporters. So these tandem reporters are constructs that, in this case, for example, you have LC3-2B, so this LC3 protein, coupled to a red and a green fluorophore. The idea behind this construct is that M-cherry is pH insensitive, so it will always be red, but GFP is pH sensitive. And what happens is when this autophagosome will fuse with the lysosome, then the GFP will be quenched, and you will only see red dots. And to give you a practical example, so this is how you would see the phagophore, how you would see the autophagosome, so they will be yellow. But then when this autophagosome will fuse with the lysosome, the pH will be lowered, and then the GFP, you will not see the GFP anymore, and you will see only red dots in this cell. And this is how we can measure autophagy flux. To give you some real examples about this, these are some cells transfected with the GFP-LC3, and you can see, you can quantify the number of yellow dots, which are autophagosomes, and the number of red dots, which are autophagolysosomes. But this is very nice because we can not only do it in cells, we can also do it in animals. And then, for example, the group of Ian Ganley and Dundee, they have made these mice that they express this M-cherry GFP-LC3. And this is a beautiful image of a retina of these mice. And you can see here, for example, in the outer nuclear layer, you see the mitochondria, but you can also see these beautiful red dots, which are labeling autophagolysosomes. And we can easily quantify the number of these autophagolysosomes by image analysis. Isn't that beautiful? Okay, so first, messages from my talk. How can autophagy be measured? So very important to measure autophagy flux with protease inhibitors or tandem reporters. Of course, as always in biology, we have to combine different methods to have the same conclusion. It's important also to use quantitative tools and can do statistics. And of course, it's very nice that you can do genetic approaches, for example, eliminating ATG5 or ATG7, and you see that you don't see this effect anymore. Okay, so now I'm going to talk a bit about selective autophagy. So I already mentioned that the cell can degrade selectively organelles, and some time ago, we thought that autophagy was only bulk, but now we know that specific organelles or cargo can be labeled for getting into autophagosomes. And I've mentioned that you can degrade ribosomes. You, of course, can also degrade lysosomes. Mitochondria. You can also degrade lysosomes. And actually, there is some idea in the field right now that maybe this bulk autophagy doesn't exist. And what happens is that many different cargo get in the same autophagosome. So this is something that we still don't know, but it could be a possibility. So the important thing about selective autophagy is how the cargo is selected. So here again, this beautiful image of mitochondria inside an autophagosome. So how this mitochondria is going to be targeted to be engulfed in this autophagosome. And regarding mitochondria is a very good example of the different ways we can get cargo into autophagosomes. So for example, you can have a direct binding of the cargo that binds to LC3, and this happens, for example, with cardiolipin in mitochondria. You can also have ubiquitin-independent cargo recognition by a receptor. And for example, mitochondria have mitochondrial proteins that bind to LC3, and this helps the formation of these autophagosomes around the mitochondria. And then we also have this ubiquitin-dependent cargo recognition, is that the cargo, in this case mitochondria, is ubiquitinated, and then there is an adapter that recognizes this ubiquitin, and then this protein, the receptor, will bind to LC3 to form this autophagosome around this mitochondria. So looking at this different cargo recognition, we can find different types of mitophagy. And to give you a little bit of background on this, the most known type of mitophagy is pink park-dependent mitophagy pathway, which is it's activated when mitochondria get damaged, and then pink, which is normally degraded in the mitochondria when mitochondrial membrane potential is stopped, then pink gets accumulated in the automitochondrial membrane, and then it's activated sparking, and then the ubiquitination of many mitochondrial proteins, and then there's a receptor like optineurin or P62 that will bind to this ubiquitin, and then it will bind to LC3, and this will form the autophagosome around this mitochondria. So pink park-independent mitophagy is the most known type. Actually, the function is to eliminate damaged mitochondria. But we have other types of mitophagy. For example, this we call receptor-mediated mitophagy, which is ubiquitin-independent, for example, via BNIP3 and NIX. So these are mitochondrial outer membrane proteins, and they bind directly to LC3 to form this autophagosome around the mitochondria. And the function of this pathway is a pathway that gets activated during cell remodeling. For example, this is a beautiful image of NIX knockout cells. Actually, those are red blood cells. And you know that red blood cells don't have mitochondria. So NIX-deficient animals maintain mitochondria in the red blood cells, and they have anemia. So this pathway is activated during cell remodeling, during hypoxia, and we have demonstrated that this pathway is very important to do metabolic reprogramming. And actually what we have found is that this programmed mitophagy of non-damaged mitochondria is very important to eliminate, to degrade mitochondria during neurogenesis of the retina. So I won't give you a lot of details, but what we have found during the neurogenesis of the retinal ganglion cells is that there is a hypoxia response that induces mitophagy, and this mitophagy is very important to eliminate mitochondria. These young retinal ganglion cells need to use glycolysis to obtain ATP, and this is very important for the maturation of the first neurogenesis of these cells. So at later time points, what we think what happened is that, so we have this mitophagy and glycolysis, and this retinal ganglion cell, of course, we all know that adult retinal ganglion cells, mature retinal ganglion cells have a lot of mitochondria. So what happens is there's a mitochondrial biogenesis. So why this complicated process? So we think that actually this change of metabolism during this neurogenesis of retinal ganglion cells is for example, to help this retinal ganglion cell to bind these very long axons, because as you know, glycolysis is making ATP in a very fast way, but it's also providing many metabolic substrates that are important for anabolism. This is why cancer cells have a lot of glycolytic metabolism. So one possibility is that this is important to have the building blocks to get this very long axon. And the second possibility, and this is just a mitochondrial quality control, because you know these mitochondria, so these retinal ganglion cells in our eyes, they need to be there for like 80 years. So it could be very important that only the good mitochondria are there, and those good mitochondria are the ones that proliferate to sustain adult life of retinal ganglion cells. So I've talked a bit about mitophagy. I give you an example. So what tools we can use to measure mitophagy? So again, we can go to this tandem technology. So instead of using LC3 coupled to GFP and RFP, the trick is to use a mitochondrial protein. And to give you an example, this is the MitoQC reporter, and there the protein is a 12 amino acid of FIS1, which is a mitochondrial outer membrane protein. So this is the MitoQC reporter, and again, the trick is the same. So mitochondria in the cytoplasm, they are yellow. So they have both colors, the green and the red. And then when this mitochondria is in the mitophagosome, still yellow because the pH is normal. But then when this mitophagosome will fuse with lysosome, the pH will quench the GFP fluorescence, and then you will see red dots. So an example in cells, so these are ARPA19 cells transfected with these MitoQC cells. So you can see this beautiful yellow mitochondria, and then when you induce mitophagy, in this case we have used a NIX-dependent way of inducing mitophagy with the ferriprone, you can see these beautiful red dots in the cytoplasm of the cell, and those are mitophagosomes. And again, we can really quantify those guys. So cells, of course, we can also do this in animals. So in collaboration with the group of Ian Galli, they have made this MitoQC reporter animal, and here you have some images of kidney and muscle. You can see that these red dots, those are mitophagosomes. So actually, in collaboration with Ian, some years ago, we have performed a map of the eye, the whole eye, comparing the mitophagy reporter with LC3, GFP, and mCherry, and compare this with the mitophagy reporter, so the FIS1, mCherry, and GFP. So we have measured many parts of the eye. So of course we are interested mainly in retina. So I give you some insights here. So this is the MitoQC. This is the autophagy reporter, and you can see again a lot of red dots in the outer nuclear layer, so in photoreceptors. The surprise came when we quantified these dots is that there is the same levels of mitophagy and autophagy, telling us that most of the autophagy activity in the photoreceptors is mitophagy. And we are currently trying to understand why mitophagy is so important in photoreceptors. Another important thing about this MitoQC is that you can fix the retinas or the tissues, for example, and then you can do also immunostaining. And here what we have done is that we have labeled these retinas from the MitoQC with conorestin, and then we have done this imaris, is a rendering, so you can see these beautiful cones, and then you can see that this is the soma and the axon. You can see that both we have red dots and free mitochondria in these cones. So again, this is very easy to do and very easy to quantify. So the questions we want to understand in my lab is, what is the role of autophagy and mitophagy in the retina? And mainly, can we find therapies based on autophagy and mitophagy that could be to use to treat retinal diseases? And I've shown you some examples of we work in development, try to understand why autophagy and mitophagy are important for development. And I'm going to give you some insights on aging, another insight on degeneration, and also some tools and some ways of manipulating autophagy. And here I'm going to tell you some examples. So the first one is we can use this MitoQC reporter animal to measure mitophagy by flow cytometry. And this is very easy because what we can do is we can isolate the retinas from the MitoQC animals, put them in culture, and then treat them with drugs for different times, and then we can dissociate these cells and then got them in the flow cytometry. And then this is the sequence that we have to get the cells, and then, for example, this is a very classical, very easy example. We have treated the retinas with control of CCCP, a classical mitophagy inducer, and you can see this is the quantification of the mitophagy high cells by flow cytometry, very easy to do. And this is just to show you that also when you do imaging of these retinas, you see more red dots in this is outer nuclear layer of the photoreceptors. So very easy to use to quantify also mitophagy by flow cytometry. Another example that we have recently used, we have used these MitoQC animals to check how this mitophagy activity is changing during development. So these are retinas from animals at different stages. Here you can see how we can quantify these red dots, but not only in mouse, but we can also do it in zebrafish. So actually what we have shown is that this is the different stages of the retina, and we have quantified both mitophagy and also the levels of oxidative stress, and we have found that in some periods of this retina, there is a peak of oxidative stress that is followed by mitophagy, and actually this is pink park-independent mitophagy. So this is mice, but we can also do it in zebrafish expressing also this MitoQC reporter. So this is in collaboration with Anne Simonsen, and we have also found that during the retinal development in the fish, there is also some levels of alterations or movement of mitophagy that also correlates with the levels of oxidative stress. I'd also like to give you some insight what we have been doing regarding aging, and for that I'll have to give you a little bit of background. So some years ago we wanted to know what was happening with macro autophagy with aging. So what we did is we used old animals, so 3 months, 12 months, and 12 months old, and then we isolate the retinas and we check LC32 levels. So if you look at these first points, this is the numbers of autophagosomes. You can see that is in the basal conditions there is no much changes in the levels of LC32, so you would say no changes in autophagosomes. However, when you do the flux, again, you can see that while young animals, you see a very nice accumulation of LC32, so a very nice accumulation of autophagosomes. Autophagosomes could not accumulate it in the aged animals. Again, telling you how important it is to measure this autophagy flux. This is just the quantification, so you can see a nice autophagy flux in the young animals and no changes in the aged animals. And we also did some qPCRs to see why this autophagy was downregulated in the aged animals, and we could find that some of the autophagy regulators, such as ATG7 and Becklin, were downregulated. Another thing that we can do to check this autophagy block is looking at P62, and this is the cargo degradation. So P62 is one of these proteins that is able to bind ubiquitin and to help the autophagosome formation, but it also gets degraded. So actually, what we could see in the aged animals is that the aged retinas have more levels of P62. But in order to be sure that this is really an autophagy block, what you have to do is also to make sure that this P62 protein is not because you have increased transcription of P62. And here you can really clearly see that, so this is the increase in protein levels, but then there is no increase in the levels of messenger RNA. So when P62 gets accumulated in the absence of increased transcription, you can really say that autophagy is blocked. And this is another way to check this autophagy, the blocking autophagy flux. Okay, so here we had demonstrated that during physiological aging in the mouse, it was at decreased levels of macroautophagy. So then we said, okay, now that we have the MitoQC reported, can we use this MitoQC reported to check what's going on with mitophagy during aging? Because we thought if general autophagy is going down, maybe mitophagy is also going down. So what we did, it was to use the MitoQC reported animals, again, using this trick that the mitophagolysosomes were in red. So we aged these animals, and then we compare the levels of mitophagosomes in the aged retinas. And the first surprise came because instead of seeing a decrease in mitophagy, we see a very nice or very important increase in the levels of mitophagolysomes in the aged neuroretina. Not only in the retina, it was the same for the RPE, so we saw increased levels of mitophagosomes in the RPE. I don't want to give a lot of details, but it's not only in the retina, it's not special about the retina, but many tissues in the body, including the brain, we saw this increase in levels of mitophagy. So that was a bit strange, so we did a transcriptomic approach. I don't want to give you the details because this is just published, but what we have found is actually that during aging, mitochondria they get damaged, and then they leak mitochondrial DNA. And we know that mitochondrial DNA is a very mitochondria are bacteria that are in our cells. So actually, when mitochondria get leaky and mitochondrial DNA gets outside, there's this C-gas sting pathway that it gets activated. Like this is a sensor pathway for pathogens. So we have demonstrated that in this during aging, C-gas sting pathway gets activated, and then you have this pro-inflammatory response. And we hypothesize that maybe these increased levels of mitophagy could be a way of these aged cells to try to get rid of these damaged mitochondria. So we thought that maybe pushing the pathway of mitophagy further could be a good therapeutic approach for these retinal diseases, for aging. So we went to look in the literature, and we found this urolithin A, which is a compound, is a postbiotic that is produced in our gut microbiota after eating pomegranate and nuts and berries. So these fruits have ellagic acid, and our gut microbiota is able to change this ellagic acid into urolithin A. And this urolithin A has mitophagy-inducing response. So we tested this urolithin A in aged animals, and again, to make a long story short, we treated the animals for 8 weeks, and we found that the animals have better neurological function, the aged animals treated with urolithin A, that they have improved memory using the novel recognition test, they have improved vision, so you can see that many of the electroretinogram waves were preserved in the aged animals. And we also saw decreased neuroinflammation. And we have also RNA-seq data showing that the CGAS-STING pathway after urolithin A treatment was downregulated. So what we found is that during aging, these mitochondria get damaged, they get leaky, they have this CGAS-STING pathway, and then this inducing this pro-inflammatory response. But this is a cytoprotective mechanism, because if you increase further this mitophagy using urolithin A, you have improved mitochondrial homeostasis, reduced oxidative stress, reduced neuroinflammation, and improved neurological function. So then we reasoned, maybe can we use this urolithin A for other neurodegenerative diseases? So in my last slide, we have used this urolithin A in the sodium iodide model. So we treat the animals with urolithin A, and we also subject them to sodium iodide, this acute retinal degeneration. You can see here that sodium iodide is reducing the thickness of the outer nuclear layer, and then the animals treated with urolithin A, we saw a very important restore, or urolithin A was avoiding this reduction in the outer nuclear layer thickness. And again, we also quantify, for example, the number of tunnel-positive cells. Sodium iodide was increasing the number of tunnel-positive cells, and it was really decreasing the number of them after urolithin A treatment. We also checked visual function, and again, urolithin A was restoring, or wasn't avoiding the decrease in vision loss after sodium iodide. So to conclude, what I've told you today is that autophagy decreases with aging in the retina, that mitophagy increases with aging in the retina and the retinal pigmented epithelial cells, that removal of aged mitochondria is essential to avoid neuroinflammation triggered by cytosolid mitochondrial DNA, and then boosting mitophagy with urolithin A alleviates A-associated neuroinflammation, and is also able to reduce, restore visual function in a model of geographic atrophy. So further questions and other things that we need to work further is what happens with the other forms of selective autophagy, what also are lysophagy pathways also activated in AMD, because we know that lysosomes are damaged in AMD. So what happens to lysophagy there? How these processes can be activated? Can we find drugs to further increase these processes? And what drugs can we use in humans in order to burst the process of autophagy? Very ambitious questions. Some further reading, so a couple of reviews on general autophagy and mitophagy, another review on lysosomes, and also I give you already the answer. Urolithin A is also promoting P62-dependent lysophagy in models of acute retinal degeneration. And if I allow, I'd just like to mention that the next ICER meeting is going to be in Valencia, beautiful, very good food, beautiful sun. It's going to be in August next year. And so actually the session proposal is open. So if you're interested in coming to Valencia, propose a session. So I'm a scientific co-chair with Paloma Litton. And with this, this is my acknowledged slide. So I've shown you data from many people. My lab was before in Spain, so many of the alumni were in CSIC. This is the current lab. And I would like to thank you for your attention, and we'll be happy to take any questions.
Jason, that's going to be a difficult question.
Jason Miller, University of Michigan. Two questions. It seems, in particular for mitophagy, that it's highly linked to mitochondrial biogenesis, right? So, although you gave a good example with RGCs, where there presumably is some temporal point where you don't want it linked to the mitochondrial biogenesis to encourage the glycolytic switch, and then biogenesis coming in afterwards. And we know the transcription factor is important for mitochondrial biogenesis, but do you have sort of more some general principles for saying, how do we predict when we're turning on mitophagy that there'll be a concurrent mitochondrial biogenesis program that's turned on versus it truly is just getting rid of damaged mitochondria and keeping sort of the levels at a lower state?
Yeah, this is a great question, because actually it doesn't make sense to just degrade all mitochondria, because then you will get rid of all of them and then the cell will have to have problems. So actually, you're completely right. There is mitophagy is many times coupled to mitochondrial biogenesis. And we have really, so in the second story where we have seen this age-leaky mitochondria, that mitophagy is good for this age-related phenotype. Of course, we wanted to check whether this was also via mitochondrial biogenesis, because you could have mitophagy plus mitochondrial biogenesis. So we went to like in vitro model to check, to damage mitochondria and check whether you needed mitochondrial biogenesis for the effects of urolithin A. And actually what we found using chloramphenicol, which blocks mitochondrial translation, chloramphenicol was not able to block the pro-survival effects of urolithin A. So at least in this model, urolithin A is inducing mitophagy, but not mitochondrial biogenesis.
Hi there. Just over to your left. Thanks very much. It's Ruben McGaugh here from Edinburgh. Lovely talk, thank you. I have a question on receptor and ubiquitin-mediated mitophagy. Do you know much if this changes as the mouse ages? Is there a switch, or are they both always active? And if I could just maybe ask one more question about, did you ever see, or is it possible that mitophagy gets overwhelmed, which is why you see accumulation of damaged mitochondria?
Yeah, two great questions. So regarding the first one, I didn't have time to give you all this data, but we have seen that in the aged retina, also in all the tissues, the pathway that gets activated during aging is pink PARC-independent mitophagy, so the damaged mitochondria pathway. So it's clear that it's just this damaged mitochondria that increases mitophagy. And regarding the second question, this is a very important question, because of course it could be do that mitochondria, so that the lysosomes are damaged and then this mitochondria get accumulated, but actually this is not, because we did flux. So of course with the tandem we are able to see mitophagolysosomes, but you can also use protease inhibitors to inject in the mice, in the MitoQC mice, and we're able to see that we could further increase the levels of red dots. So actually we could still go further. So the activity is not blocked, we can go further. And this goes in agreement with the idea that boosting mitophagy with urolithin A is also cytoprotective. So there is room for improvement, and there is hope for aging, actually.
Thank you.
Hi, Daniel Haas from Emory. You showed a couple different kinds of autophagy, organelle-specific. Is there a favorite kind that a starving cell will do?
Yeah, that's also a very good, important point. Actually, so starvation was the classical way to induce autophagy, and people thought that just randomly the cell was getting a bit here and a bit there. But actually this is not the case. So actually, depending on the timing of starvation, people have done proteomics and have checked that at the beginning you get more or less like bulk and some mitochondria, but if you prolong time, there are some substrate that are spared from these autophagosomes. And actually, at the beginning, you get a bit of mitophagy, but at the later time points, you don't get mitophagy. So the kinetics is very important, and tell us that during starvation or prolonged starvation, there are pathways that control what cargo is going to be degraded.
Beautiful talk. RP cells never undergo starvation because they're always eating photoreceptor outer segments. And in fact, they use part of autophagy machinery to digest those outer segments. So what is unique about autophagy in RPE cells, and how does that change with age or not?
Yeah, actually this is a good idea. So as you mentioned, some of the autophagy machinery is important to degrade this photoreceptor outer segment in this non-canonical autophagy called H3-associated phagocytosis. So there you get LC3 in single membranes around these phagosomes. So actually, this is one of the process by which autophagy proteins get activated, but you can also use drugs. For example, when we use urolithin A in RPE in culture, we see activation of autophagy. So actually there are many different stimuli that can get this autophagy going on in RPE cells.
I have a question.
Go ahead, you first.
The beautiful pictures. Is it only my impression that the red dots are not really in the inner segments, but they are sort of on the edge of the outer nuclear layer, in the outer nuclear? Not in this picture, but we looked at the mitophagy pictures.
Yes.
And does that mean that a mitochondria sort of decides that it wants to get mitophagized and moves up in the retina?
Yeah, this is a very good question, actually. We have seen, because for example, I didn't have time to show the data, this is unpublished, but we have crossed the MitoQC reporter, for example, with the RD10. And actually, in the RD10, we see the accumulation of these red dots in this upper part of the retina. So we are trying to see or to think whether the transport is also important. And this could also be related where lysosomes are, because you need lysosomes in order to get fusion. So in the RD10 animals, we also have shown that lysosomes are not working very well. They have lysosomal membrane permeabilization. So maybe in this case, only lysosomes that are working are in the upper part, and the other ones are damaged. But this is just, we need to really demonstrate that. Yeah.
So two questions, and one of them is very much related to what Florian just asked, so thank you, Florian. That was good. So segue for this. So it looks like mitochondria would have to be transported to where those lysosomes were, right? Are you seeing fragmentation of mitochondria first and then transport of these fragmented mitochondria, or activation of some sort of damaged response to mitochondria? Those mitochondria to be specifically transported to lysosomes.
Yeah. Well, you're asking a very difficult question because this is in vivo retina. So we need to find out a way of getting these retinas lyses, put them on the microscope, and check mitochondrial movement. So this is something that we have started in collaboration with some groups in Alicante. But yeah, it will be really nice to know whether they're fragmented first and they migrate afterwards, or whether this is at the same time. So those are very difficult experiments, but they are very, very informative.
And the second question is, it seems to me that in the retina, these highly metabolic demanding tissues, the mitophagy and mitochondrial repair and biogenesis, as has been pointed out, has to be quite robust, right? I mean, in order to live this long and under this much stress and pressure from metabolic stress, mitophagy has to be a really robust program, right, in these tissues, right? Where do you really think that therapeutic interventions are in sort of targeting that mitophagy or repair? Where do we really need to focus our attention?
Yeah, that's a difficult question. Actually, I didn't answer you asked me whether the mitochondria can, too much mitophagy could be bad. So actually, I think that we really need to carefully measure how much we stimulate the pathway, because if we go for too much, we're going to get rid of all these mitochondria. So actually, the way I see this is either we use treatments that are intermediate, so we give some burst and then we wait, and then we another one and then we wait, or we have to have molecules that have like a very slow release, for example. Yeah. And this will be also true for treatment with general autophagy inducers. It's not good to have all the time autophagies in a bit of the way of this caloric restriction and restricted feeding. You don't have to start all the time. You have to start a little bit, and then you need to recover. You need to do protein synthesis again. So we have to really think how we are going to administer those drugs when the day comes. Yes.
Okay. Thank you.
Thank you. What's in fish that induces mitophagy?
We think this is developmental mitophagy in a similar way what we have seen in the mouse. So we think that during retinal development, what we have shown, at least in the mouse, in the fish, we have not demonstrated that, is that you have a hypoxia peak, and this hypoxia peak is triggering this NIX-dependent mitophagy, and this is important to get rid of these mitochondria to change the metabolic profile. So.
Yeah, Osha, I was wondering if you fell asleep.
As a clinician, I'm just being educated so much. I've got so many, as I say, my brain buzzes when I listen to all you guys. So does the mitochondrial DNA haplotype influence how these mitochondria undergo mitophagy?
Yeah, this is a great question. I don't know, but it would be very interesting to really check whether the haplotype will be influenced. But I cannot answer that. Very good.
And DNA mutations.
Yeah, of course. Yes.
Thank you very much. Excellent.
Thank you.
So I hope you all enjoyed this as much as we did. I think I have enough material to write at least 3 grants for BrightFocus that I hopefully will be intelligent. And no, I really, really appreciated the different aspects that we got thrown into, and I think it was, for me, it was really great. I hope it was the same for you.
So just thank you very much to the 9 speakers for giving us such informative talks, and to BrightFocus for sponsoring the session. So thank you. Thank you all.
And I want to say a couple of things. One is that we had an amazing session, and thank the speakers for that. But really, Florian and Sarah, you guys did an amazing job of picking the speakers and actually getting them on board with the program and really adapting to what we're trying to accomplish here. So I really thank you both. This has been an outstanding session, and you guys are responsible for that, so thank you. And again, thank you, BrightFocus. This has been really an important partnership, and I really am terrific. I'm thrilled to have such a terrific program and opportunity here. So thank you very much.
I'll just say, pick up a T-shirt, and that's the end of the day. I'll close it. Have fun.